Thank you so much. So I'll get started. Welcome to the ARO seminar series centered around the topic of auditory brainstem and midbrain. And today we are delighted to have Dr. Michael Roberts as our speaker. My name is Marina Silveira, and I'm assistant professor at the University of Texas at San Antonio, and I'll be your moderator for today's webinar. But before we start, if you have any questions during the presentation, please put them in the chat. But after the talk, feel free to virtually raise your hands, unmute yourself, and directly ask the questions. Any questions that are not addressed during this session will be answered by Michael in a follow-up email. Closed captioning for this meeting is available, and this section will be recorded and will made available for additional viewing for people who could not attend. If you have any technical difficulties with Zoom, please email headquarters@aero.org, and they will be able to assist you. Okay, so after that, today it really is my great pleasure to introduce Dr. Michael Roberts. He is an associate professor in the Kresk Hearing Research Institute at the University of Michigan. Michael received a BA in biology from the University of Chicago in 2000, and then he completed a PhD in cell and molecular biology at the University of Texas at Austin in 2005. After that, he did 2 postdoctoral fellowships, and they were both focused on the neural circuits in the auditory brainstem. His first postdoc was with Larry Trussell at the Volo Institute, and the other postdoc was with Nace Golding at the University of Texas at Austin. Michael started his lab at the Kresk Hearing Institute at the University of Michigan in 2015. His lab is focused on determining how neurocircuits in the auditory midbrain support sound processing in health and disease. So when Michael started his lab, which is crazy to think that's 10 years ago, we knew very little about the cell types and classes of neurons in the inferior colliculus and how to use molecular markers to identify these neurons in the auditory midbrain. But in the last decade, his lab has made many new discoveries that really lead towards how to identify these classes of neurons in the midbrain. And those were very important discoveries for our field, so we can make new advancements since we can now identify those different classes of neurons in the inferior colliculus. So all this research that came from Michael's lab, that I'm sure he will share a lot of it with you today, really has had a significant impact in the auditory field and opened many new research questions for many other labs in the field. And on a personal note, Michael, he is an excellent mentor. You can double check him with any of his trainees in his lab, and I'm sure they'll say the same thing. One of the best choices that I made was to be a postdoc in Michael's lab for 6 years, and I can clearly say that he's one of the best mentors I ever had. And in addition, he's also a great colleague to have around, and I'm sure everyone in Kresk will confirm that as well. But other than doing all this great research in his lab, Michael is also the director of the Hearing Balance and Chemical Sensing T32 training program at the Kresk Hearing Institute. And without further ado, it really is my pleasure to introduce Michael and welcome Michael, who presented his work that's titled Determining the Structure and Function of Microcircuits in the Inferior Colliculus.
Well, thank you so much, Marina, for that really kind introduction. And thank you to Kathy and the other members of the ARO Seminar Committee for giving me this opportunity today. This is a really exciting time for me to share a talk with the ARO community, because as Marina mentioned, we're 1 month away now from reaching the 10-year anniversary of my lab. So it's a fun opportunity for me to pull together various threads of things we've been working on over the years to kind of give, hopefully, what will come across as a big picture of what has been a driving force problem we've been focused on for the past decade. So go ahead and jump right in here. So very broadly speaking, what really motivates our work is trying to understand at a mechanistic level how neural circuits detect and encode the various features of speech and other vocalizations that are important for us to make sense of speech and other vocalizations. And also, how do auditory circuits change following hearing loss? So, of course, to understand how things change after hearing loss, we first have to also understand how things work under normal conditions. And we're very interested in my lab in studying this in this brain region called the inferior colliculus, or IC. You will hear me say IC probably a million times today. So if there's one acronym to know, IC would be a good one. So just to quickly orient everybody, I've got here on the left two different images of brains that show you where the IC is. This top image, I believe, is from a rat, and you can see the IC labeled here. The inferior colliculus sits just rostral to the cerebellum and just behind the superior colliculus. In many mammals, it's actually covered by cortex, but conveniently in mice where we work, the inferior colliculus is actually right on the surface of the brain. There's no cortex over it. The IC, you can see in a rodent brain, is a pretty big part of the brain. You look in a human brain, I don't always show this image because of this. It looks kind of underwhelming. It's this little bump here, down here sitting on the surface of the midbrain brainstem. Okay, so here's this brain region. Why do we care about it? Well, to me, one of the most fascinating things about the auditory pathway is once sound information enters the brain from the auditory nerve and cochlea, it goes to the cochlear nucleus and then is sent to a whole host of brain regions throughout the brainstem, not only in the cochlear nucleus, but also in the superior olivary complex, which is comprised of numerous nuclei, the lateral lemniscal nuclei, which has 3 different nuclei. So you have all this quick dispersion of information. This code gets sent to different specialized processing centers. But then something really interesting happens. Almost all the ascending output from the auditory brainstem, from the cochlear nucleus, superior olivary complex, and lateral lemniscal nuclei, all this output comes together in the inferior colliculus in the auditory midbrain. So we often call the inferior colliculus the hub of the auditory pathway for this reason, because just like a hub airport is an airport where lots of flights come in from all over the place and go out to all over the place, the inferior colliculus has a similar setup. Almost all sound information converges on the inferior colliculus, where it's processed before it gets sent up to the auditory thalamus and eventually to the auditory cortex. So in this circuit diagram here, all these green arrows are summarizing, very simplistically summarizing a lot of the ascending auditory pathway. It's important to mention the inferior colliculus is also a hub from the standpoint that it gets a lot of descending input from the auditory cortex, and the inferior colliculus itself sends descending input to various parts of the brainstem. So just from this gross circuit diagram, you might get the idea that the IC is playing a very important role in the auditory pathway, and it's very important to understand how it works. And there have been dozens and dozens of studies over the years studying what the IC's role in hearing is. And we could spend hours just on this alone, but to very broadly summarize, we know the IC is important for speech and vocalization processing. It plays important roles in sound localization, also important roles in echo suppression, and in some species, echolocation, notably in bats, but also some humans have been able to echolocate, although we don't know if they actually need the IC for this, but I bet they do. And more broadly, we think of the IC as being important for identifying behaviorally relevant auditory cues or signals, signals that have some really importance for the survival of an organism. If we look more closely at the structure of the inferior colliculus, there's been a lot of work on this over the decades, but I think the landmark studies were really from Doug Oliver and Kent Moress back in the early 80s, where they looked at the cat inferior colliculus. And they found that there are 3 main subdivisions of the inferior colliculus. There's the central nucleus that you can see here. The central nucleus is the target of most of the ascending auditory input from or output from the brainstem. At the top of the inferior colliculus, there's the dorsal cortex. This receives a lot of descending input from the auditory cortex. And then on the side of the inferior colliculus is the lateral cortex, sometimes called the external cortex. The lateral cortex is a fascinating subregion that also, in addition to auditory input, gets a lot of somatosensory and multisensory input. Most of what my lab's been focused on is related to the central nucleus of the IC, because I'm personally very interested in just the basic ascending sound processing pathways. But you'll see that we also touch on some of these other subdivisions later. So, like most of the structures, if not all in the ascending auditory pathway, the central nucleus of the IC is tonotopically organized, kind of in a funny way along this diagonal, such that low sound frequencies are best represented more dorsally and laterally. And as you proceed down and towards the midline, higher sound frequencies are represented. This structure is so clean that we actually can see things called isofrequency lamina or sheets in the central nucleus of the IC. So the neuronal structures, so many of the neurons are oriented in a way that you can actually pick out different layers that represent different tonotopic lamina in the central nucleus of the IC. We also know from really nice work from Gerard Borst's lab and Dan Yano's lab and some others that the lateral or the dorsal cortex of the IC has some tonotopic organization. The lateral cortex of the IC does not seem to have a clear tonotopic organization. IC also does a lot of interesting things beyond just simple sound processing. So there are a number of studies, including this one from Anna Chambers when she was in Dan Pauly's lab, that show that the IC exhibits plasticity. So this study that I really like showed here that in neural recordings in control animals, a classification algorithm could really reliably determine from neural activity when different speech tokens were presented to the mouse. Then, when the auditory nerve was really decimated, over 95% of spiral ganglion neurons were destroyed with Wabane treatment. 7 days later, you can see this neural code got really degraded. But by a month later, this neural code had largely been restored. And so this suggests a lot of plasticity taking place in the IC and other parts of the auditory pathway. So I think it's very important to keep in mind that what the IC does is probably very susceptible to change with time. And then one of my favorite things about the IC, but also I think the biggest mystery about the IC, is that it's very, very rich in intrinsic circuitry. And I think if you have one take-home message from today, I'd like it to be that you think about the fact that the intrinsic circuits in the IC have immense computational power, and we've barely scratched the surface of how they work. So some really nice stereology work from Randy Kaleza in rat showed that the inferior colliculus contains about 5 times as many neurons as the rest of the auditory brainstem nuclei combined. So that right there, just from the sheer number, I think that tells you a lot about there must be a lot of processing going on in the IC. I'll tell you a lot more about this in a second, but we know that IC neurons can be divided into numerous molecularly defined neuron types. I wouldn't be surprised if we end up just thinking there are 15 to 20 different neuron types in the IC at least. So far, we and others have found markers for some of these molecular markers like VIP, neuropeptide Y, cholecystokinins, and somatostatin. It's also really well established from work from Doug Oliver, Manolo Malmierica, Wallace and Palmer, and others, many others, that most IC neurons have local axon collaterals. So this is an interesting arrangement. There are a lot of principal neurons in the IC, or neurons that project beyond the IC. In fact, it's not clear there are any true inner neurons in the IC that don't have projections that leave the IC. But at the same time, these projecting neurons have local axon collaterals. And as I'll show you more in a minute, we've seen this with some of the neuron types we've identified here. These are all local axons with potential synaptic release sites of VIP neurons in various parts of the inferior colliculus. We see the same thing with MPY neurons. A couple of reconstruction examples from Wallace and Palmer's work showing in black here these beautiful axonal arbors of single in vivo labeled neurons from the IC. So these axons are meant to impress you that there must be a lot going on with the circuits within the IC. So a lot of computational potential is sitting right there in the IC. So very broadly speaking, long-term goal of my lab is to determine how neural circuits in the IC detect the spectral temporal features of speech and other vocalizations. And as we learn more and more about that, we also want to determine how these IC circuits change in hearing loss and hearing disorders. But as you can imagine, and we know very well from other brain regions, if we want to accomplish these goals, the first thing we have to be able to do is probe the neural circuits in the IC. And to do that, we have to be able to reliably identify and manipulate specific classes of IC neurons. But this has been a big problem that the field has been stuck on for a long time. So a lot of the rest of the talk today, I'm going to talk to you about what classes of neurons make up the IC and how are IC microcircuits or local circuits organized. So as kind of a teaser of where we're trying to get with this, I put up some circuit diagrams from different brain regions where neural circuits have been characterized in much more detail than we currently have for the IC. So I think probably few places in the brain, if nowhere, have been more well characterized than the hippocampus and cerebral cortex. And we know a tremendous amount now about how different neuron types connect to one another, interact with one another to perform computations, and even how specific neuron types support learning and behavior. Similarly, this has been done well in olfactory bulb, and even within the auditory pathway, some parts of the auditory pathway have lent themselves very well to these kinds of analyses using more conventional approaches. So the dorsal cochlear nucleus being a notable example, where we know all these different cell types and how they interconnect with each other. So what do we know about the local circuitry within the IC and the cell types in the IC? So one of the most fundamental observations is that in the central nucleus of the IC, there are two main morphologies of neurons. There are so-called disc-shaped cells, labeled in this diagram with D. And the notable thing about the disc-shaped cells is they keep their dendrites in narrow bands that run parallel to the isofrequency lamina of the IC. So you can think of the disc-shaped neurons as being neurons that probably have pretty narrow tuning to sound frequency because their dendrites tend to run along one isofrequency layer. In addition to disc-shaped cells, there are stellate cells that send their dendrites in multiple directions and often send them across these tonotopic layers in the central nucleus. But the problem is, even knowing about disc-shaped and stellate-shaped cells, subsequent studies, especially from Doug Oliver's group, have shown that both of these neuron classes can come in glutamatergic and GABAergic varieties, so they can be excitatory or inhibitory. And there's a wide variety of different physiological and anatomical properties for neurons in both of these groups. But we still think this distinction is very helpful and important. So beyond this, a lot of different studies have been done to try to classify the neuron types in the inferior colliculus. So people have used a variety of approaches like anatomy and neurochemistry, in vitro electrophysiology, so this would be brain slice electrophysiology, and in vivo electrophysiology. And time again, what comes out of these studies, which are really well done, is that there's almost a continuum of neuronal properties in the IC. So it's very hard with conventional techniques relying only on anatomy or electrophysiology measures to identify a distinct class of neurons in the IC. So to give one example, for a while it was thought that IC neurons could be defined based on their frequency response areas, so how they respond to pure tone stimuli, and that these frequency response areas should have one of three main shapes. And then this really comprehensive study from Adrian Reese and Alan Palmer showed that this is really kind of messy because there are a whole host of different frequency response area shapes that fall along a continuum. And so determining dividing lines becomes really tricky. So this is the big theme in the IC literature for a long time has been the IC is a very diverse place where it's very difficult to robustly and reliably categorize neuron types. So for the rest of the talk, I'm going to focus on how we've been addressing this problem. We've got three main sections. In the first section, I'll talk about identification of VIP and MPY neurons in the IC. And as we go along, I'll have some pictures and names and places. So a lot of the work I'm about to tell you about was actually pioneered by Marina Severa, our host today, and also a previous postdoc in the lab, David Goyer. We also have a really long-standing, wonderful collaboration with Brett Schofield and his lab, which at the time included a lot of tremendous work from his postdoc, Nicole Beebe. So let's jump into the identification of neuron types. So when I started my lab, our basic hypothesis was that we will be able to identify neuron types in the IC only if we combine information from a variety of domains. So instead of focusing primarily on anatomical markers or physiological markers like many past studies have done, we thought it was really important to combine examinations of neurons across numerous modalities. And I think the biggest innovation is we really started to focus on molecular genetic markers for neuron types. So many of you will probably appreciate that molecular markers for neuron types have been extremely successful and powerful approaches in other brain regions, especially cortex and hippocampus, but many others. So we wanted to harness the power of modern molecular approaches in transgenic mice as ways to identify and manipulate neuron classes, then look at the properties of these neurons using brain slice electrophysiology, morphological analyses, looking at the axonal projections, where these neurons get input from. And in work I'm not going to tell you about today that we're really getting going on now is how these different neuron types respond to sound. So one way to think about this is really like a taxonomy problem. If you're going to identify a new species of animal, you want to know many different features about that animal. And so that's our approach here, is how do we find patterns across these many different feature spaces. So where to start? We decided to start looking for molecular markers among neurons that express different neuropeptides. So neuropeptides are small proteins that are anywhere from 3 to 70 amino acids long, roughly. They're really evolutionarily ancient signaling molecules. So in fact, they were among the first neurotransmitters. They precede fast neurotransmitters we think about today, like glutamate and GABA. You can find neuropeptide signaling all the way back in nervous systems of Cnidaria. Also, neuropeptides are exciting because in many cases, they're very important neuromodulators in different parts of the brain. And we know from other brain regions that neuropeptides are often good markers for neuron types. So we screened a lot of neuropeptides that might be good markers for neuron types in the IC, largely using the Allen Institute's in situ hybridization database, looking for interesting expression patterns in the IC. And the first one we landed on is vasoactive intestinal peptide, or VIP. And so we were very fortunate that there was already a VIP Cre mouse. So in this mouse, Cre recombinase is expressed in VIP producing neurons. We can cross this Cre mouse with an AI14 mouse, which is a TD tomato reporter mouse, so that only cells expressing Cre, so in this case only VIP cells, should remove this stop cassette and lead to expression of TD tomato. So when we have VIP Cre by AI14 mice, we should have red fluorescent VIP cells. And indeed, when we obtained these mice and bred them together, we were really, really happy to see when we looked in the inferior colliculus, VIP neurons here shown in magenta are located throughout the inferior colliculus. We're going from, these are coronal sections viewed from the caudal to rostral. And you can see VIP neurons aren't found evenly everywhere. They're more common in the dorsal cortex of the IC, but they're also present in the central nucleus, and they get sparser and less common as you go more rostral in the IC. We subsequently did in situ hybridization to determine that, to find that VIP neurons are glutamatergic. So here you can see in this magenta label for VIP mRNA, these VIP positive cells in 90-some% of the cases also express vesicular glutamate transporter 2, indicating they're glutamatergic. Our mouse line driving TD tomato expression also labels many, but not all, of the VIP expressing neurons. So it's a good tool. So already we're getting a hint VIP neurons are an excitatory neuron type. We made patch clamp recordings in brain slices from VIP neurons, and when we do the recordings, we can fill the cells with biocytin, which allows us to go back and recover the morphology of the neurons later. When we did that, we found that VIP neurons have a stellate morphology where their dendrites project in multiple directions. And hopefully it comes through over Zoom. You can see we've got these diagonal gray lines meant to indicate very roughly the direction of the isofrequency lamina in the IC. And so you can see that these neurons in the central nucleus of the IC do not follow the laminar shape. And so we believe that these are stellate cells. So we believe VIP neurons are a type of stellate cell. Marina, when she joined my lab, found another marker, our second marker for a neuron type in the IC, and this time it's neuropeptide Y. Neuropeptide Y there are a number of mouse lines that use neuropeptide Y to label neurons, and the first one we worked with that was really useful is this MPY-HR-GFP mouse, in which GFP fluorescence expression is driven by the MPY gene. And so here you can see in these serial sections from the IC that MPY neurons are found throughout the IC. And our collaboration with Brett Schofield's lab, this was very nicely plotted here. You can see MPY neurons in the three main subdivisions of the IC. When Marina did in situ hybridization, she found that these MPY neurons do in fact express MPY mRNA, and they express the vesicular GABA transporter, so they're GABAergic neurons. So we're happy at this point. VIP neurons are glutamatergic, MPY neurons are GABAergic. It turns out MPY neurons are also stellate. They send dendrites in multiple directions, not running usually anywhere near parallel to the isofrequency lamina. Our next big test was, we know from past work and from our own work shown here that if you just randomly record in brain slices from IC neurons and inject current steps to depolarize the neurons and hyperpolarize them, you see a whole host of different firing patterns and hyperpolarization response patterns. And so again, this theme of there's a diversity of responses and properties in the IC emerges. So we're very excited when we target whole cell current clamp recordings to just VIP neurons or just MPY neurons. In both cases, the vast majority, 97% of the neurons, have these very simple sustained firing patterns. So when we depolarize the cell with increasingly large current steps, you can see the VIP neurons and also the MPY neurons fire very consistent trains of spikes. So we go from this diversity of firing patterns to when we have a molecular marker to target a specific group of neurons, we see one specific firing pattern almost exclusively. We've also, because we have these mouse lines, we can now start tracking where these neurons project to, which can be another indicator of what constitutes a neuron type. And so first for VIP neurons, because we had a VIP Cre mouse, we were able to use a Cre-dependent adeno-associated virus, or AAV, to drive GFP expression only in IC VIP neurons. So we did intracranial virus injections in the IC, and then looked all over the brain for where there would be VIP neuron projections. And again, this is work we did in close collaboration with Brett Schofield's lab. So this was, I think, one of the most surprising things to come out of our studies so far, is that this one group of VIP neurons, as a population, projects to most of the targets that are known for IC neurons in general. So within the inferior colliculus, the VIP neurons project to all the three main subdivisions in the ipsilateral IC. They also project to the three main subdivisions in the contralateral IC. They send descending projections to the nuclei of the lateral lemniscus, the ventral nucleus of the trapezoid body, and the dorsal cochlear nucleus, so all over the auditory brainstem. They send ascending projections to the nucleus of the brachium of the inferior colliculus and the three main subdivisions of the medial geniculate, which is the auditory thalamus. In fact, we're really struck when you look in the ventral portion of the medial geniculate, the main lemniscal portion, we often see that VIP neurons make these really large, beautiful boutons, suggesting they have very large glutamate release sites in the medial geniculate. VIP neurons also project to some other non-auditory brain regions, which is really fascinating. So these include projections to the periaqueductal gray, which is often involved in escape and defensive behaviors, to the superior colliculus deep layers, where you have audio and visual integration, intercollicular tegmentum, this small cholinergic nucleus in the brainstem, the LPGI, and even to this dopaminergic nucleus, the subparafascicular nucleus. So this suggests this population of VIP neurons in the IC can broadly influence a lot of processes across the brain. We've also, with Marina, looked at where MPY neurons project. So for this, because we didn't have a Cre mouse line at the time, which we do now, we used retrobead labeling. So Marina injected retrobeads in the medial geniculate and then saw that they often labeled neuropeptide Y neurons in the IC. You can see co-labeling here with retrobeads. So this indicates that MPY neurons form part of the IC's inhibitory projection up to the auditory thalamus. Marina, at the time, a very talented undergraduate who's now a graduate student at Northwestern, Justin Anair, did another nice retrobeat project where they showed that MPY neurons project to the contralateral IC. So they project across the commissure of the IC, suggesting they play a role in binaural inhibition. And there was a really striking pattern here where MPY neurons send their projections to the contralateral IC over a larger area than non-MPY neurons, suggesting almost like a surround inhibition kind of modality. So in summary, in finding VIP neurons and MPY neurons together, we think they represent about 2/3 of the stellate neurons in the central nucleus of the IC. VIP neurons account for about 5% of glutamatergic neurons in the IC, possibly more since the VIP mouse doesn't actually label all the VIP neurons. So it seems like a small population, but I'll note that VIP neurons and cortex account for 2 to 3% of neurons in the neocortex, and yet there are dozens of really impressive studies about how important VIP neurons and cortex are for behavior and other computations. Neuropeptide Y neurons account for about 33% or a third of the GABAergic neurons in the IC, and about 7.5% of all the neurons in the central nucleus of the IC. So all told, these two markers of neuron types have allowed us to make a lot of progress, and we're starting to get a little bit of a handle on what the neuron diversity in the IC is like. So now I want to spend some time telling you about some discoveries that being able to identify and manipulate specific neuron types have empowered us to make. So this next section is going to focus on some physiological measures of local circuits in the IC. And again, a lot of this work was done by Marina in combination with Ioani Herrera, a talented graduate student in the lab, and our former postdoc, David Guyer. So first of all, Marina was very interested in neuropeptide Y as a signaling molecule. And so she found that the Y1 receptor for neuropeptide Y is expressed by about 80% of glutamatergic IC neurons. So in this in situ hybridization, if you look in B here, the cyan color VGlut2 mRNA is labeling glutamatergic neurons. And you can see many of these glutamatergic neurons express the MPY Y1 receptor, so type 1 receptor for neuropeptide Y. We also had a Cree mouse that expresses, when we cross it with a reporter mouse, expresses TD tomato in the Y1 receptor expressing neurons. And so this gave us access to these neurons that express the Y1 receptor, which happen to account for about 80% of all excitatory neurons in the IC. So with this, we started to do some optogenetic circuit mapping. So Marina injected a virus and an AAV into the inferior colliculus of mice, of the Y1 receptor Cree mice, to express a channel rhodopsin, in our case, either Chronos or soma-targeted chrome, in the Y1R positive neurons. And then she went and targeted recordings in brain slices to either Y1R positive neurons, which are going to be 80% of your glutamatergic neurons, or Y1R negative neurons, many of which are probably GABAergic. Then she can flash light to activate the Y1R neurons transfected with the virus and drive synaptic release onto the Y1R positive or negative neurons. And so Marina found here in these nice current clamp recordings that the Y1R positive neurons routinely project onto other glutamatergic Y1R neurons. You can see here, nice EPSP. They also routinely project onto Y1R negative neurons. So we think this means that Y1R positive neurons often project to GABA neurons as well within the local IC. So this is starting to map out some of the local circuitry within the IC. Then a really interesting thing Marina found was that if she put blockers of fast inhibitory neurotransmission, so gabazine to block GABA A receptors and strychnine to block glycine receptors, if she put these in her bath solution, recorded from a neuron while activating Y1R neurons with optogenetics, she would often get these really interesting responses where a very brief pulse of light, which hopefully you can see here, this very narrow blue bar that's only representing about 2 millisecond light stimulation, would often cause trains of excitatory input, sometimes firing, that lasted to 300 milliseconds in some cases. So what would cause this, we think, is that Y1R neurons and probably other excitatory neurons within the IC are very tightly interconnected, have a very high connection probability to one another. So that if we block inhibitory synaptic transmission and activate a handful of excitatory neurons, that leads to this recurrent activity where other excitatory neurons continue exciting each other and driving synaptic input to the recorded cell for dozens of milliseconds. This was also evident in voltage clamp recording, where you can see these downward deflections representing inward current that continue for some time. This all fits with something else we know about the IC, which is that the IC is the site of initiation of most audiogenic seizures. So seizure activity or epileptiform activity is generally a result of highly interconnected excitatory circuits within a brain region. And so it turns out the IC has these kind of connections, and this study really looked at this functionally for the first time. And then a really exciting thing to come out of this is because these neurons express the Y1 receptor, Marina next asked, well, what if we activate the Y1 receptor with an agonist, LuPro-MPY? What's that going to do to this recurrent excitation we see here in this control trace? And so it's known that the Y1 receptor is a GPCR, G protein coupled receptor, that's coupled to GO. So it's an inhibitory receptor. And so when Marina activated the Y1 receptor, you can see that that inhibited most of that recurrent excitation that we saw here on the left. So this suggests that neuropeptide Y signaling in the IC might play an important role in regulating excitability, potentially especially during periods of hyperexcitability. So these are interesting things to think about in terms of what happens with central gain in hearing loss and what happens in audiogenic seizures. So we're just starting to scratch the surface with this, and Marina and her lab's really following up and chasing a lot of these leads down. So another thing, mapping local circuits with functional approaches in the IC, Marina found that neuropeptide Y neurons provide inhibitory input to other neurons in the local IC circuit. So here you can see examples of recording from a random IC neuron, activating neuropeptide Y neurons with channelrhodopsin, and you can see an inhibitory postsynaptic potential here, or a train of them when Marina stimulates with a train of light pulses. So this suggests that neuropeptide Y neurons also inhibit other neurons in the local IC. We've also looked beyond just the local circuits in the IC. We've started to look at some of the ascending circuits into the IC, and these have also intriguingly revealed some things about local circuits in the IC. So back when David Goyer was in the lab, he expressed the excitatory opsin chronos in the DCN, dorsal-cochlear nucleus, using viral injections, and recorded from VIP neurons in brain slices. And he would often see an interesting response to a light pulse that looked like this. You have, in current clamp, an excitatory depolarization that's followed very rapidly by an inhibitory potential here. And so we saw this and we thought, this really looks like we've got direct excitatory input from the DCN, followed by some local source of feed-forward inhibition. And in fact, when David put on the GABA A receptor blocker gabazine, he saw that the excitatory portion of the response here that we saw under control conditions got prolonged, and the inhibitory portion of the response went away. So because David was doing this in brain slices where all the other inputs to the IC were severed, and he was just directly activating DCN terminals with light, we think this is the circuit that these data propose, which is that DCN provides excitatory input directly to VIP neurons, but also to some local inhibitory neuron that then provides feed-forward inhibition to the VIP neurons. So you can imagine this is a really cool circuit motif because this feed-forward inhibition truncates the time course of the excitation, so may have a very important role in temporal processing in the IC. Our wonderful graduate student, Ioana Herrera, has been looking at another projection pathway to the IC, in this case from T stellate cells in the ventral cochlear nucleus up to the IC. So again, she's injecting a virus into the VCN to express the excitatory opsin chronos in T stellate projections to the IC. When Ioana records from MPY or VIP neurons, she often sees excitatory depolarizations rapidly followed by inhibition. So it's cleaner here in this VIP example. And again, this points to a feed-forward inhibitory circuit where the excitatory T-stellate input impinges both directly on the MPY or VIP neuron, but also onto an inhibitory interneuron in the IC that provides feed-forward inhibition. So from all these different streams of evidence, we're starting to put together a admittedly still very rudimentary circuit map of what's going on in the IC circuits. So here I've drawn our MPY neurons, which are GABAergic, VIP neurons, which are glutamatergic, and then some generic glutamatergic and GABAergic neurons. And we know VIP neurons from anatomy must have local projections. We don't know to which other neurons yet. We know that MPY neurons project to many of the local glutamatergic neurons and also to the VIP neuron, most likely because they're glutamatergic, but we don't know that for sure. We know that ascending input from the AVCN from T-stellate cells impinges on both MPY and VIP neurons, but also drives feed-forward inhibition to these cells, so must contact another GABAergic neuron population, possibly MPY neurons themselves. DCN fusiform cells also project to VIP neurons and drive local feed-forward inhibition in the IC. So you can see we're starting to get some sort of connections worked out here. So in addition to just who connects to whom and how strong their synaptic input is, another important thing when thinking about computations in a brain region is how do those synaptic strengths change over time? So there's a very well-known phenomenon in synaptic physiology called short-term synaptic plasticity, where it's known if you repeatedly stimulate a synapse, some synapses get weaker with time, others get stronger with time with repeated stimulation. So these are called synaptic depression or synaptic facilitation. So we've also been looking at how inputs to IC neurons change during trains of stimuli. So Marina found in her work with the Y1 receptor positive neurons that their synapses onto other neurons in the local IC tend to not have very strong short-term plasticity. So sometimes they would get a little weaker with time, sometimes a little stronger with time. But overall, if we summarize here, these lines stay pretty close to 1, suggesting not very strong short-term synaptic plasticity in these local circuit elements. In contrast, when Ioane looks at short-term synaptic plasticity in the T-stellate inputs to the IC, she sees really marked short-term depression. So here's a T-stellate input to an MPY neuron, and you can see during this train of stimuli, the average response gets smaller. Similarly, with a VIP neuron, the average response gets smaller. When Ioane quantifies this by measuring paired pulse ratios, ratios less than 1 indicate short-term depression, and you can see overall at both MPY neurons and VIP neurons, the T-stellate synapses undergo short-term depression. So this is interesting because this suggests that when you have a T-stellate cell input to the IC, so for example, a sustained sound is driving T-stellate input, the T-stellate input's going to be strongest initially and then get a little bit weaker with time as the stimulus continues. So you can imagine this has important implications for adaptation to sounds and how ascending input might affect computations in the IC. Ioane's also looked at short-term plasticity at a variety of stimulus frequencies and sees that short-term depression gets, if anything, a little bit stronger as she goes to 50 or 70 hertz stimulus trains. So all of this suggests that the synapses in the IC themselves are also important sites of computation that we need to keep in mind when thinking about the computational potential of the IC. Skip this real quick in the interest of time. So finally, I want to end with a quick overview of some new approaches we're taking to mapping local circuits in the IC. So I showed you a bunch of work in this last section using functional approaches to mapping local circuits, but we also want to really figure out and nail down the anatomical tendencies of axon projections for specific neuron types in the IC. So here's the problem. The IC has incredibly dense circuits. So we always make a big deal when we talk about the IC, as I did in this introduction, that the IC is a hub of integration. The IC gets all sorts of ascending inputs, descending inputs, and also has a tremendous amount of local axon projections within it. So because of this intermingling of all these different projection pathways, it's very, very difficult using conventional approaches to track the axons of individual neurons unless we use a labeling approach that labels one neuron at a time. But as you can imagine, labeling one neuron at a time is very slow and tedious and very low throughput. Another problem is that the axonal projections of most IC neurons have very complex three-dimensional shapes, and these shapes are often lost or very difficult to accurately reconstruct if we use conventional anatomical approaches where the IC is cut into sections. So to overcome this, Eli Uez in my lab, who's a very talented graduate student, has been taking what we think is a really exciting and powerful approach. So the main goal here is to use sparse neuron labeling. So we only label a small percentage of neurons at a time. This is kind of like a classic Golgi labeling approach, but using modern methods. And then we want to, in addition to the sparse labeling, be able to image the axons of neurons in the intact IC without cutting brain slices. So to do this, we use sparse labeling using this mouse line called Morph3. Morph3 mice use this really cool genetic trick where, first of all, there's a reporter construct that is expression of it's blocked by a stop cassette that can only be removed in Cre-expressing neurons. So we have the LOXP sites here. But then the open reading frame of the reporter protein, in this case, it's the spaghetti monster fluorescent protein, is preceded by a repeat of 22 cytosines. So 22 cytosines puts the open reading frame of the reporter gene out of frame. It's essentially like a frame shift mutation, so this gene won't actually get expressed. However, in a small percentage of cells, when this gene is duplicated during mitosis, there will be a frame shift error restoring this to 21 cytosines, allowing the reporter protein to be expressed in a very small portion of cells. So this Morph3 mouse is a really cool way to get sparse labeling in Cre-expressing neurons. Eli's also been figuring out and optimizing approaches to clear the brain through delipidation and to immunolabel intact brains using electrophoretic approaches. And then he takes his samples to the light sheet microscope, which we have very fortunate to have a really fantastic Zeiss light sheet in our microscopy core here. And he's able to get images like I will show you now. So here's an image from a VIP Cre by Morph3 mouse. So only a small percentage of the VIP neurons are expressing this spaghetti monster fluorescent protein, which actually in the Morph3 mouse is no longer fluorescent. So we use an antibody to label the neurons expressing this. And the labeling works really well. So you can see a number of VIP neurons here. And to orient you, the IC is turned a little bit on its side here. So dorsal is actually kind of going over here, and the midline is up over in this direction. So if I advance now, we should see the movie here. So it's hard to appreciate just how amazing these data sets are until you really get to play with them in a computer where you can rotate things in 3 dimensions. So I'm hoping these movies help give you a sense of what we're able to capture with this light sheet and sparse labeling approach. Okay, so you can already see just with this handful of VIP neurons, there's a lot going on. So there's a lot to figure out. We're going to zoom in a little more. And this also illustrates why we need sparse labeling, because even with sparse labeling, we can actually reconstruct neurons with this, but it's still pretty complicated. This movie gives you a zoom in with a higher magnification, showing you even in a completely intact IC, we can detect axonal boutons, these little beads on a string looking things. We can see dendritic spines, you can see here. So really remarkable resolution that's allowing us to start to reconstruct VIP neuron local axon projections. Let's let this zoom in a little more here. The detail is just really amazing. Okay. So Eli, with help from one of his undergraduate researchers, Lila Aragdi, has started to reconstruct VIP neurons and their axonal projections in one of the brains he's collected so far. So you can see some of these neurons, you can see, for example, in yellow here, You can see the dendrites very well, and then the axons spread out. And in fact, some of these reconstructed axons are crossing the midline of the IC, which is roughly over here, projecting all the way to the contralateral IC. So if I animate this movie, you'll get a little bit better sense. You can also see a number of cell schemata that we still need to reconstruct the neurons processes. But hopefully this gives you a sense that we can start to capture the morphology of the VIP neurons. And now what we're starting to do is more quantitative analyses to see what are the axonal projection tendencies of VIP neurons. So here's a single VIP neuron. You can see its dendrites coming off the soma. Then this long, thin process is an axon. And if I rotate this in 3 dimensions, you'll see that the neuron itself is very planar in its dendritic field, but its axons going out in multiple directions. And then if we go to the next slide, we have 2 more VIP neurons constructed, and these 2 send axons through the commissure of the IC to the contralateral IC. So you see their axons all the way over here. So there's a blue neuron and a green neuron. And we really believe with this approach and the right quantitative analyses, we'll start to be able to pick apart for different neuron types in the IC, how they tend to project. Okay, now go ahead and cut this off. So that wraps up what I want to tell you about today. So I'll just quickly summarize that we've identified VIP and MPY neurons as molecularly identifiable classes of IC stellate cells. So to us, this is really exciting because we and others now are starting to get a handle on some of the molecular markers for neuron types in the IC. We found evidence the IC is really rich with recurrent excitatory circuits, and that these circuits can be modulated by neuropeptide Y signaling. Ascending input from the cochlear nucleus, both the VCN and DCN, can elicit feed-forward inhibition in the IC. So these circuits I told you about where like a DCM projection excites both a VIP neuron and an inhibitory input to the VIP neuron. We've shown that ascending inputs from the T stellate cells in the ventral cochlear nucleus undergo short-term synaptic depression, which has important implications for how the IC processes input from the VCN. And finally, we're really excited that our new sparse labeling and light sheet microscopy approaches are going to provide really powerful ways to map local circuits in the IC. And so finally, I'd just like to thank many wonderful members of our lab. A lot of the work today I showed you is from Marina Severa, David Goyer, Ioane Herrera, and Eli Uez. And we have a number of wonderful collaborators, and much of the work I showed you today was also in collaboration with Brett Schofield in his lab. And especially in these times, I'd like to give a huge shout out and thanks to our various funding sources over the years. So with that, I will stop and be happy to take any questions.
Hello, Michael. Thank you so much for your presentation. It was very informative. And I have a question here. Of course, you did a great job in like showing different, like, NPY and VIP, but I kind of thinking this kind of separation. I don't know if you ever attempt to study the crosstalk between them, especially one of them is glutamatergic and the other is GABAergic. And I have in this concern, I have 2 questions. Number one, by studying them or merging the study of them in the same mouse, do you think both of them share the same isolamina? I know that like they are distributed in isolamina and or stellate, I mean cross lamina, but they share the same like the same area, or everyone is structured in a specific area? So this is number one. Number two, I don't know if, like, based on question number one, do you think everyone has its own tuning based on this distribution? For example, do you think the GABAergic neuron here can depict or draw the frequency area of the other one or not? So basically, what is the crosstalk between the GABAergic and the glutamatergic neuron here? And thank you.
So I don't know that we've actually looked in the same sections at VIP neuron and MPY neuron localization, but by just looking at where they're located in the IC, they have very overlapping patterns. I think there are more MPY neurons in the central nucleus than there are VIP neurons, at least higher density, but I don't think they're segregated in any way. I think you would certainly find MPY neurons near VIP neurons. Our looks at the axon projections, though, also suggest neurons don't need to be neighboring to be connected. We think actually the connectivity between neurons is probably extremely common in the IC. And so to answer your first question, I don't think there's anything that really separates where VIP and MPY neurons would be found in the IC. For the second question, we absolutely think that the local circuitry is playing a very important role in shaping things like frequency response areas of IC neurons. There's past physiology work with pharmacology that suggests that, but we'd really like to get in there and be able to do this in a cell type specific way and say, for example, if we're making an in vivo recording and we inhibit MPY neurons in the IC with something like optogenetics or DREADS, does that change how other neurons respond to sounds? And I would be shocked if it did not change how they respond to sounds. Great questions.
Thank you so much.
Pat,
Patrick?
I think you're next.
Yeah.
Hey, Michael. Great talk. Good to see you. So I've had a question for some time, I think, about inhibition within the inferior colliculus. And I tend to think about having, we'll say, different functional roles, like, right? So maybe an oversimplification, but for the sake of conversation, say, inhibition needs to shape sound responses of neurons within the lamina. It needs to perform sideband inhibition. Some of the data you guys are showing too here today is it needs to limit recurrent excitation within the structure, but also provide feed-forward inhibition into the thalamus as well. And so with the idea of breaking down into different cell types or cell groups within the inferior colliculus, it'd be very nice if they like segment, right? So it's like you have a molecular marker for an inhibitory neuron that does one role, or even a morphology that performs one role and a different one that performs another role. It's starting to seem maybe that that's a naive interpretation, and that it's probably more likely that you have subsets that perform some of all of these functions. But I guess kind of an open question of, based on everything you guys see, are you able to kind of inform this model? Like, are there subsets of, like, is MPY more important for some of these functions, or are they doing a little bit of everything to where there's probably, you know, inhibitory neurons are doing multiple roles functionally?
Yeah, that's a huge and very great question. It's a question that keeps us up at night a lot. And I think, I mean, to be perfectly candid, we're just scratching the surface at the functional implication of all this. And Brett Schofield and I have spent a lot of time talking about what if it turns out that IC neurons, like in a group like neuropeptide Y neurons, don't just have one functional role. And in fact, at this point, I'd be shocked if they have just one functional role because they constitute so many neurons. So our leading idea is that maybe what's more important about them is who they connect to. I suspect we'll eventually find that MPY neurons, VIP neurons, other neuron types can be subdivided into groups that project to different targets. And maybe that makes them act, you know, perform a specific role in those circuits. So if we think about them rather in terms of doing a specific thing related to sound processing, instead doing a specific thing related to a specific circuit, like this neuron might be involved in providing feed-forward inhibition, and then it can be recruited by a number of different sounds in different ways, depending on where the neuron's located, to provide feed-forward inhibition to accomplish different computations. That might be another way to think about it. We've called that like the circuit widget hypothesis. So shifting the framework from what's it doing for sound to what's it doing for a circuit. But that's really, a lot of that's speculation at this point. One of the big problems in dealing with neuron type identification, there's always the lumper and splitter debate of, we could probably keep splitting these neurons, and probably will at some point split them more into smaller and smaller groups. And a lot of that's going to depend on what's really useful for us in trying to understand computations related to hearing. So we're just very early stages at making sense of a lot of that.
Thank you.
We have one more question in the chat, but it's already 12. But if you want to comment really quickly, so does your map of the circuit so far gives any sense of the functional role of the IC? For example, whether these inputs are combined in a discrete classes or continuum. So I think build this up on Pat's questions.
Yeah, it was very similar to Patrick's question. But great talk. Thanks.
Yeah, thanks, Luke. I mean, I will add, one of the things I mentioned earlier, the study from Randy Kaleza that showed that the IC has about 5 times more neurons than the auditory brainstem. So one way to think about that, when you have cases in the brain where you go from fewer neurons to a huge increase in the number of neurons, that's often referred to as an expansion layer. And there are a number of cases in the brain where you have situations like that. Probably, one of the biggest cases being the inputs to the cerebellum onto the granule cells. That's an enormous expansion layer. You go from comparatively few inputs to granule cells in the cerebellum, making up the largest neuron type in the brain, essentially. So what happens often in these expansion layer circuits is they're helpful for separating out features. And part of that is you have neurons that integrate inputs from a lot of different places. And some neurons, possibly through experience, learning, things like that, are going to respond better to certain combinations of inputs. So it might be the case that we end up finding the IC is really an array of neurons, each of which tends to respond better to different combinations of inputs. And that might help with the feature separation that's taking place in the IC. But that, again, that's speculation.
Okay, thank you, Michael. Do you want to say something?
Well, I was just wondering, and so do you think that will end up being the combination in more discrete classes or more continua, like the FRA finding, that it seems like maybe there's not such clear-cut classes? Or is
Yeah,
it too
so I
early
think,
to tell?
again, I think where the discrete classes is going to come in is if we think of them less in terms of how they're processing sounds per se, like in a single unit level in vivo, but more in terms of how they're contributing to circuit operations. And so I think it's a little bit of kind of a frame shift in how we consider what's going on. And I think people in other areas like in neocortex are much more used to thinking that way. And then think how the biological, the sensory problem gets put onto this circuit when they already know the circuit functions in a particular kind of way.
Okay, that's great. Thank you so much, Michael, for this great talk and everyone that contributed with questions. And thank you everyone for attending today and being here at the ARO seminar.
I have a question. Right.
I guess that's okay.
So,
Okay, go for it.
Michael, great talk. Do you think that inferior collicular has a role in computing and coding salient sounds? The salience has a significant role.
Yeah, in fact, I think I had a great conversation with Carl Candler once. I don't know if Carl's here and if he'd want me to say this out loud, but he said, if you're a mouse, the IC might be all you need, right? And I think you look evolutionary, the IC is really ancient structure. It existed before the tympanic ear. You find it all the way back in teleos fish and in frogs called the Taurus semicircularis. And so I think it's evolutionarily probably very important structure for doing things with sounds that were important for survival. And probably in humans, we've co-opted it more and more for, things we care about like speech, music, whatever. But it's really remarkable to see in all these different species across evolution, the IC repeatedly being used for processing sensory stimuli that aren't even always sounds, that sometimes are just stimuli that have rapid temporal information in them, like the weakly electric fish use the torus semicircularis for amplitude modulation detection of the electric fields and things like that. So it's a fascinating structure that I think probably has a lot of importance for those more evolutionarily old tasks than human speech perception.
Okay, thank you.
Thanks, Jose. Thank you, Marina.
Yeah, thank you so much, Michael. And thank you everyone for showing up and make this and contributing with questions.
Great. Take care, everyone. See you soon.
Okay. Bravo.
Thank you, Jose.
Really good.
Bye.